You know, if you've ever tried to swat a fly that just seems to know exactly where your hand is going before you even move it.
Oh, yeah, it's completely impossible.
Right. Well, you have a tiny inkling of what it's like for scientists trying to design a vaccine for HIV. You build a weapon, you take aim, and by the time you fire, the virus has already changed its shape.
It really is the ultimate shapeshifter.
Exactly. It's so frustrating.
And, you know, the sheer math of that shapeshifting is what makes it so elusive. For decades, the virus has basically been winning this evolutionary arms race against our best scientific minds.
Because it mutates, right? Like incredibly fast.
So fast. It mutates so rapidly that traditional vaccine approaches, which rely on showing the immune system a static, unchanging picture of a virus, they just fail completely against HIV.
But what if you could trick that shapeshifter?
Yeah.
What if you could like back it into a corner where in its desperation to escape your attacks, it accidentally drops its shield?
And reveals its absolute most vulnerable weak spot.
Yes. That shift in strategy is exactly what we are looking at today. Welcome to today's deep dive. We aren't just talking about a new chemical compound here.
No, we are talking about a monumental shift in how we approach virology entirely. We're turning the virus's own survival instincts against it.
If you've ever wondered why we still don't have an HIV vaccine, today we're bringing you the answer, or rather a massive leap forward in finding one.
It's a huge leap.
We're unpacking a groundbreaking 2026 paper in the journal Science, led by Ashwin Skelly, that provides a literal molecular blueprint for how to outsmart HIV.
And the core of this paper outlines this brilliant two-step mechanism. It essentially forces the immune system to create the exact weapons needed to fight the virus.
Which represents the culmination of, years of trying to solve one very specific problem.
Yeah, years. The problem of how we generate broadly neutralizing antibodies.
Right. Okay, so let's unpack this. Our mission for this deep dive is to really understand this two-step trick and how scientists engineered a virus to map it all out.
But before we get to the trick itself, we probably need to talk about the weapon.
Yeah, we need to talk about these broadly neutralizing antibodies, or bNAbs as they call them.
Right. So to understand why bNAbs are considered the holy grail of HIV research, you have to look at the mechanics of how HIV survives inside a human host.
Which is pretty messy from what I understand.
Oh, incredibly messy. When HIV replicates, its genetic copying machinery is just, well, it's really sloppy. It makes constant genetic typos.
So inside a single infected person.
The virus is constantly churning out millions of slightly different versions of itself, yeah.
It's like changing the locks on your house every 10 minutes. The immune system works incredibly hard to forge a key, but by the time the key is made, the lock is entirely different.
That is the perfect way to visualize it. Standard antibodies act like those outdated keys, but there is a very rare class of antibodies, these bNAbs, that can neutralize a massive variety of these viral strains all at once.
Wait, how do they do that if the virus keeps changing?
They manage it by completely ignoring the rapidly mutating parts of the virus. Instead, they target what we call the conserved regions.
Okay, so by conserved, we mean the parts of the virus's architecture that it absolutely cannot afford to change.
Exactly. It's the structural load-bearing wall of the virus.
Because if the virus mutates that wall, the whole viral structure collapses, right?
Right.
And it can no longer infect cells.
That load-bearing wall is exactly what the researchers went after here. Specifically, the paper focuses on a microscopic area called the V3 glycan patch.
The V3 glycan patch. Let's give you guys a mental image of this architecture. Imagine the HIV virus is a sphere covered in these protruding spikes.
Those spikes are the envelope glycoprotein, or N for short.
NVEF, right. Now, HIV doesn't just leave those spikes exposed. It covers them in this dense forest of sugar molecules, which biologists call glycans.
And what's fascinating and kind of terrifying is the origin of those sugars. The virus actually steals those glycans from our own host cells to build its camouflage shield.
Wow. So because the shield is made of our own body's sugars, our immune system looks at the virus and thinks, oh, that's just a normal part of me.
Exactly. It creates immunological tolerance. It's an incredibly effective invisibility cloak.
But every cloak has a seam. And that seam is a tiny patch at the base of what's called the V1. No, wait, the V3 loop, right?
Yes, the V3 loop.
Right. And for those of you who love the deep molecular details, this patch involves specific sugars anchoring at positions N332 and N301 on the viral protein sequence.
And the N there just stands for asparagine, which is the specific amino acid where the sugar attaches itself.
Ah, okay. And tucked right in there is a tiny critical sequence of amino acids called the GDIR motif.
The GDIR motif, that is the engine of infection. That specific sequence helps the virus bind to the human cells it wants to invade. It is essential machinery.
So it literally cannot change it.
Right. The virus physically cannot mutate that GDIR sequence without destroying its own ability to spread.
So antibodies that manage to bypass the dense sugar forest and lock onto this V3 glycan patch, they must be powerful.
Unimaginably powerful. We know they're capable of neutralizing up to 65% of all circulating HIV strains globally.
Wow. Okay, let's do a quick recap here to make sure we're mapping this out clearly for you. HIV covers itself in a stolen sugar shield, but it has one unchangeable weak spot, the V3 glycan patch, which is required for infection.
Right.
And if we can target that patch, we can stop 65% of all HIV strains. So the obvious question you're probably screaming at your device right now is, why can't we just make a vaccine that teaches the body to target this patch?
It's a great question, and it comes down to the grueling mechanics of how these rare antibodies naturally develop.
They do develop in some people naturally.
They do, but in a very small fraction of human patients. And it is not a fast process. It takes months or even years of constant active HIV infection for these to organically develop.
Because the virus mutates, the immune system adapts, the virus mutates again to escape.
Exactly, and the immune system has to stretch and contort to keep up. It's a long, exhausting evolutionary dance happening inside the patient's lymph nodes.
And we obviously can't give a healthy human being an active replicating HIV infection and wait 2 years hoping their immune system eventually figures out the dance steps.
No, definitely not. We need a shortcut.
So finding that shortcut required watching the dance step by step. But from a purely research perspective, scientists had a massive roadblock, didn't they?
Yeah, we didn't have a reliable animal model to study this evolutionary process.
It's like trying to figure out a complex magic trick when you're only allowed to see the final reveal, the rabbit pulled out of the hat once every 2 years.
Right, you never get to see the sleight of hand. You can't observe the setup or the misdirection.
So to reverse engineer the magic trick, researchers needed to be able to watch it happen consistently, like in slow motion from multiple angles, tracking every single molecular movement.
Which brings us to the outbred animal model. We needed a subject with an immune system wonderfully complex and diverse, much like humans. So researchers turned to rhesus macaques.
But macaques don't naturally catch human HIV, right?
No, they don't. So the scientists had to rig the deck. This introduces the concept of an evolving immunogen.
An evolving immunogen. Let's break that down.
So they engineered a specialized hybrid version of the virus called a SHIV, a simian-human immunodeficiency virus. Specifically, a variant called SHIV.5MUT.
SHIV.5MUT. The evolving immunogen part is crucial for you to grasp here. Usually when you get a vaccine, like a flu shot, you are getting a dead or inert piece of a virus. It just sits there.
Right, it doesn't change.
But this engineered SHIV actually replicates inside the macaques. It provides a high viral load, and most importantly, it mutates. It actively fights back against the monkey's immune system over the course of a year.
The experimental design was brilliantly simple. They infected one group of macaques with this highly specialized SHIV.5MUT. Then they infected a control group with the standard wild-type version of the virus, known as BG505.N332.
Okay, so engineered virus versus standard virus.
Exactly. And over the next year, they took continuous blood and lymph node samples to track every microscopic battle in this war.
And the results they published are just staggering. I mean, if you read a lot of scientific literature, you're used to seeing incremental improvements.
Yeah.
A 5% bump here, a minor correlation there.
But this was different.
This was a sledgehammer. Yeah. Out of 22 macaques infected with the engineered SHIV.5MUT, 14 of them developed incredibly potent V3 glycan, broadly neutralizing antibodies within the very first year.
And the potency of these antibodies cannot be overstated. The team isolated 12 distinct lineages of antibodies from these macaques that achieved up to 68% neutralization breadth across a massive 130 virus test panel.
That is wild.
To put that in perspective, they performed on par with the absolute best, most elite human-derived antibodies ever discovered.
And the control group, the 14 macaques infected with the standard wild-type virus, zero.
Zero.
Not a single one produced these elite antibodies in that time frame. 15 to 0. But reading through the source material, I noticed a detail that seems completely counterintuitive to me.
What's that?
The genetic difference between the standard virus that failed and the SHIV.5MUT virus that succeeded is microscopic. It's a difference of just 4 amino acids in a region called the V1 loop.
But just 4 amino acids, yeah.
I struggle to see how changing just 4 tiny building blocks suddenly turns these macaques into bNAb-producing factories. What exactly changed in the architecture of the virus?
Well, to answer that, we have to look at the central discovery of this paper, which they call the two-step mechanism.
Ah, okay, the two-step mechanism.
Because they were taking continuous samples from the lymph nodes over that year, they could sequence both the viral mutations and the antibody developments in real time.
They were finally watching the sleight of hand in slow motion.
Exactly. And here's where it gets really interesting. Step 1 is the decoy.
The decoy, okay.
When the macaques were initially infected with the engineered SHIV, their immune systems immediately noticed that V1 loop on the surface of the virus.
Because of those 4 specific amino acid changes you mentioned.
Yes. Because of those changes, the V1 loop was highly exposed and highly recognizable. It acted like a massive red flag waving in the wind.
So the immune system took the bait.
It took the bait and poured all its resources into creating standard, everyday antibodies to attack this V1 loop.
But we know HIV is a shapeshifter. It doesn't just sit there and take the hit. When the immune system starts destroying the virus by grabbing that V1 loop, the virus has to adapt.
And that leads to step 2, which is the trap.
The trap.
To escape this massive immune attack on its V1 loop, the virus is forced to mutate its own structure. It starts churning out what virologists call escape variants.
Okay, so it alters its own DNA to physically shorten the V1 loop, right? By pulling it back inward.
Crucially, yes. And as it shrinks that loop, it drops several of the bulky sugar molecules covering it. The paper refers to this process as becoming hypoglycosylated.
Hypoglycosylated. Let's use a visual analogy to really cement this mechanism for you. Imagine the HIV virus is a heavily armored tank rolling through the immune system.
Okay, I like this.
The V1 loop is like a huge, obvious target painted on the side of the tank. The immune system is the defending army, and it starts firing everything it has at that obvious target.
Right, and eventually the tank takes a hit and gets a flat tire. The initial wave of standard V1 antibodies is the artillery taking out the tire. The tank is now compromised.
So to survive and keep rolling, the tank crew has to fix the tire. They pop open the heavy armored hatch on the top of the tank to climb out.
But the moment they open that hatch...
Exactly. The moment that the virus purposefully shortens its V1 loop and drops its sugar shield to escape the first attack, they accidentally expose the incredibly vulnerable, soft interior of the tank.
And waiting for that exact moment of vulnerability are the immune system's snipers.
Yes. The interior of the tank is the V3 glycan patch, and the snipers are the rare bNAb precursor cells. By trying to escape the first attack, the virus accidentally physically exposes its own Achilles heel.
The V1 loop was literally covering the V3 patch. When the virus shortened V1 to save itself, it pulled back the curtain.
That is just incredible molecular judo.
It really is. The mechanics of this physical exposure are what biologists call removing steric hindrance.
Steric hindrance. What does that mean in plain English?
It's essentially molecular crowding. Imagine trying to plug a USB cable into your laptop, but there is a massive, bulky plastic case blocking the port. You just can't get the angle right.
Oh, I've had phone cases like that. It's the worst.
Right. So the long V1 loop and its thick sugar shield act like that bulky plastic case, physically blocking the rare bNAb precursors from attaching to the V3 patch.
But when the virus shrinks the loop, it removes the plastic case.
The port is suddenly wide open.
And once that port is open, those rare precursor B cells finally wake up, lock on, and start maturing into the elite special forces we need.
And the researchers provided fascinating evidence of how these antibodies physically overcome the remaining shield. They noted that the successful antibodies in the macaques utilized a wide range of approach angles and had CDRH3 loop lengths ranging from 14 to 25 amino acids.
Okay, we definitely need to translate that jargon. What is a CDRH3 loop, and why does the length matter to the listener?
Think of the CDRH3 loop as the grappling hook of the antibody. A standard antibody has a relatively flat binding surface, which is useless against HIV because the virus is covered in those protective sugars.
Right, the glycan forest.
You need a way to reach through the canopy of the sugar forest to grab the viral surface beneath. The CDRH3 loop is a protruding molecular arm that reaches through the gaps in the sugars.
So the 14 to 25 amino acids is literally just a measurement of how long their grappling hooks are.
Precisely. And the fact that the length varied so widely across 12 different lineages is profoundly important.
Why is that?
It proves that creating these elite antibodies isn't just a 1 in a billion genetic lottery ticket. The macaques were using lots of different genetic starting points, different lengths of grappling hooks, different approach angles to get the exact same neutralizing result.
If you expose the target clearly enough, the immune system will figure out a way to hit it using whatever tools it happens to have in its toolbox. So the scientists deliberately tricked the immune system into forcing the virus into a corner, where the virus's only escape route required it to commit evolutionary suicide by exposing its own weak spot.
It is a breathtaking piece of rational design. But, the ultimate goal of mapping this complex evolutionary dance in macaques was never just to cure monkeys.
Right, so what does this all mean for you? How do we take this incredibly intricate year-long observation and turn it into a human vaccine? Because we obviously can't inject humans with a live, replicating, engineered SHIV virus just to start the two-step dance.
No, that's far too dangerous.
So how do we translate it?
If we connect this to the bigger picture, the absolute beauty of having a complete molecular map of this two-step dance is that we don't have to wait for it to happen organically. We don't need the live virus to act as a decoy and execute step 1.
Wait, really?
We can just skip directly to step 2.
We just build the tank with the hatch already open.
Yes. The paper lays out a very specific 3-stage blueprint for a human vaccine regimen based entirely on the cheat codes they extracted from the macaques.
Okay, let's walk through this blueprint.
Stage 1 is the prime. You inject humans with highly engineered, safe, protein-based immunogens. The researchers call these the del4 or del8 variants.
And what makes them special?
These are static viral proteins where the V1 loop has already been artificially shortened in the lab, and the bulky sugars have been removed.
Oh, wow. So you were showing the immune system the exposed V3 glycan patch from day 1. You are artificially removing the steric hindrance so the USB cable can plug right in.
Exactly. This immediately wakes up the snipers, priming those rare bNAb precursor cells across the board. And this highlights a critical debate in the HIV field between lineage-based design and lineage-agnostic design.
Let's define those quickly.
Traditional lineage-based design is like trying to forge a vaccine that works for one very specific genetically defined naive B cell. It's like forging a key for one specific lock. If a human patient doesn't have that exact genetic lock, the vaccine fails.
Which is a huge problem because human genetics are incredibly diverse across the globe.
Exactly. What Skelly and the team are proving here is the power of a lineage-agnostic approach.
Because they're just blowing the door off the hinges.
Right. By maximizing the physical exposure of the vulnerable patch, the vaccine doesn't care what specific genetic starting point you have. It can grab any capable B cell in the vicinity. It is a much more robust strategy for protecting a diverse global population.
So stage 1 primes the immune system by leaving the door open. But waking up the precursors isn't enough, right? I mean, they have to get stronger. I'm assuming this is where stage 2 comes in.
Stage 2 is the boost. You come back weeks later and boost the patient's immune system with new variants of the viral protein. But these variants are special. They contain 10 very specific key genetic mutations alongside further modifications to that V1 loop.
I have to ask about those 10 mutations. Why are they so important, and what exactly are they doing mechanically to the immune cells?
Those 10 mutations were lifted directly from the macaque data. By watching the virus and the immune system co-evolve, the researchers identified the exact structural changes the virus made that successfully forced the antibodies to become elite neutralizers.
So it's like a training program.
Mechanically, these 10 mutations act like a molecular gym. They subtly change the surrounding architecture of the virus, creating barricades that make it slightly harder for the baby antibodies to bind.
So the antibodies are forced to adapt. They have to stretch, contort, and mutate their own grappling hooks to keep a grip on the target. You are putting them through a targeted workout regimen to build their strength and affinity for that GDIR motif we talked about earlier.
It is guided evolution. You prime them with the open hatch, and then you boost them with the exact structural barricades needed to turn those raw recruits into elite special forces.
Okay, so if stage 1 wakes them up and stage 2 puts them in a molecular gym to build their strength, I imagine stage 3 has to be the final exam. You have to eventually test if they can fight the real thing in the wild.
That is the exact philosophy behind the third stage. The final boost utilizes a cocktail of naturally occurring, highly diverse HIV envelopes sourced from all over the world.
The final exam.
Right. This ensures the antibodies haven't just become hyper-specialized at fighting the engineered lab vaccine. It proves they can recognize, latch onto, and neutralize the chaotic, messy diversity of actual HIV strains you would encounter in the real world.
And to give you all a sense of how fast this science is moving, it isn't just theoretical whiteboard math. The source material emphasizes that immunogens utilizing these shortened glycan-deficient V1 loops are actively entering human clinical trials right now.
Yeah, trials like HVTM-307 and HVTN-144.
They are testing these exact forced unshielding concepts in human volunteers as we speak.
It is a thrilling time for immunology. We are finally moving away from the blind trial and error that characterized the last 40 years of HIV research and moving into an era of rational, structure-guided design based on a complete high-resolution map of the evolutionary battlefield.
Okay, let's do a final recap of the journey we just went on. For decades, scientists knew that rare, powerful bNAbs could stop HIV, but they couldn't figure out how to generate them because they couldn't watch them develop in real time.
Right.
So they engineered a specific simian-human virus, SHIV Put5-MUT, and infected macaques. By taking continuous samples over a year, they discovered a two-step trick.
The decoy and the trap.
Yes, when the immune system attacks a decoy target on the virus's surface, the virus is forced to mutate and shrink that target to escape. But in doing so, it drops its sugar shield and accidentally exposes its own crucial weak spot, the V3 glycan patch.
And having captured this entire dance on a molecular level, scientists extracted the cheat sheet. They are now using that data to design a 3-stage human vaccine that skips the decoy phase entirely, forces the immune system to recognize the exposed weak spot, and puts the resulting antibodies through a molecular gym to guarantee broad protection.
And what's really vital for you to take away from this deep dive is that this represents a fundamental paradigm shift in how we approach vaccination entirely. This isn't just a story about HIV anymore.
No, it's not.
It's about mapping the genetic dance between a host and a pathogen and using the pathogen's own survival mechanics against it.
We are learning the rules of viral evolution so we can break them. By understanding that a virus's defensive shield often hides a structural dependency, we can engineer ways to force the pathogen to unshield itself.
Which leaves us with a truly provocative thought for you to mull over. If we can map out this forced unshielding trick for something as notoriously evasive, heavily armored, and rapidly mutating as HIV, how far can we push this molecular judo?
That's the big question.
Think about oncology. Many aggressive cancers survive by coating themselves in a dense sugar shield, very similar to HIV's glycan canopy, tricking the immune system into ignoring the tumor. Could this exact lineage-agnostic, forced unshielding approach be used to strip the camouflage off cancer cells, forcing them to expose their vulnerabilities to our immune snipers?
It definitely forces us to ask, how many seemingly invincible diseases are just one flat tire away from popping the hatch?
Just waiting for the sniper. That's all for today's deep dive. Stay curious.